which is a fluorescent emission device using red acid. My name is Suarez. This is Hayley, James, and Chris. These are allies. And the main objective of our device was to design and construct a device that will be capable of obtaining a KD measurement of fluorescent proteins by simply taking pictures of that fluorescent protein pair and then processing those pictures using a MATLAB code and obtaining a KD measurement from those pictures. Our design was broken down into three main parts. The first part being obtaining the KD measurement using the flexion of fluorescent proteins. The second part being obtaining, again, the KD measurement of a fluorescent protein pair, however, this time using an infrared microscope. And the third and most important part was obtaining a KD measurement of fluorescent proteins, however, this time using our own imaging device that we created and designed. And at this point, you guys might be asking yourself, how were we able to accomplish this? Well, there's this physical phenomena by the name of red, which happens when two fluorescent proteins are found within close proximity of each other, in this case, 10 nanometers or less. And in this case, the first fluorescent protein being CY-PET, the first fluorescent protein gets excited at 414 nanometers, which then emits a first 75 nanometers, and that emission of the first fluorescent protein excites the second fluorescent protein, which in this case is Y-PET. And Y-PET, as you can see in this diagram, emits at 530 nanometers, therefore showing a red signal. Right underneath the diagram, you can see the mathematical model of red, and right underneath the mathematical model, it's two graphs showing red signal.
The first method we used to calculate KD was using the fluorescent. We had a 384 well plate, which we had our proteins. How the fluorescent would work would be, it would excite the proteins for each sample as the wavelengths to be specified, which was the 414 and 475 nanometer wavelengths. And the fluorescent would return the emission values at the different wavelengths. And the wavelengths we were most interested in were the 475 nanometer and 530 nanometer. Once we have the emission values at those peaks, we could calculate the infrared value. And once we have the infrared values for each sample, we could take those values and run them through a software called Prism. And in Prism, we would use this equation to calculate the KD measurement. And before we started work on our imaging device, we wanted to see if it was even feasible to calculate a KD value using imaging data. So after we ran our assay through the fluorescent, we would take a picture of each well using the Nikon Eclipse TI microscope. The microscope had 3 filter cubes, which corresponded to the excitation and emission wavelengths we're interested in. Once we had the images, we would run each through the MATLAB code, which would return the image intensity value. As you can see on the left here, it's an image from the well. And then once we had the values from both data sets, the image intensity and the relative fluorescent unit values from the fluorescent, we normalized both data sets and plotted them. For our assay, we made sure that the CY-PET band gap concentration was held constant, while the Y-PET UVC9 concentration varied. And as you can see in these graphs, the imaging data and the fluorescent data correlated very well. And once we found that the two correlated, we ran both data sets through the Prism software to determine the KD. From the fluorescent, we got a KD value of about 5.34 micromolar, and the imaging data returned a KD value of about 3.64.
So finally, the last method was imaging-based detection of fluorescent signals using our imaging box. And that figure over there was a SolidWorks model of how we wanted our imaging box to look like. We needed it to have a closed box for a dark room, so it prevents outside light from obscuring our data. And we needed LEDs to excite the samples at 414 and 475. We used a UV LED and a blue LED, respectively, at those wavelengths. And we also needed emission filters so that we can isolate the wavelengths that we wanted. So on the first shelf over there, we placed the samples. You can either put them in a 384 well plate or a microfluidics chip. On the second shelf, we have on the second shelf we have the filter, and on the filter we put the LEDs, which would excite the samples on the top right here. And finally, on the bottom, we had our camera, which would take images of our samples. And after 20 weeks, this was the final design of our imaging box.
So this is an image we actually captured with our imaging box. This is an image of whitehead fluorescing when it's been expanded at 475 nanometers. As you can see, this is a blank well, so you can definitely tell the difference as to when there is or there is not protein. All of these wells also have different concentrations of C whitehead. So Chris is going to help me out with a little demonstration of how our box works. On that first level is where our sample is. It's a well plate. We have little markers in there made out of paper. Paper fluoresces under light, so that's how we're able to position our sample how we want to. We need 3 values from our imaging box. We need to measure FLDD, which is the emission wavelength of C whitehead when it's excited at 414. We need FLAA, which is the emission wavelength of whitehead when it's excited at 475, and we need M total, which is whitehead when it's been excited when it's in proximity with C whitehead, so that's infrared filtering. Right now, the filter we have in there is the 530 emission filter, so we're able to get the FLAA and the M total values. To get the FLAA value, we'll turn on the blue light because that is a 475 nanometer range, and then to get the M total value, we turn on the UV light, which excites at around 414 nanometers. And then when we were ready to go and get the other measurement we needed to FLDD, we would switch the filter and we would excite it with the UV light again and get our FLDD. And then we'd go look at our little paper markers that we had in there and move it on to the next sample that we needed to do. And we were able to get 9 samples at a time, which was fast and easy. When we were running this with a microfluidic system, which you guys hopefully see the chip that's being passed around, there's little holes on the side of this box. I'm not sure if you guys can see them, but we ran our tubing through that. And we had our syringe pump on one side going into the inlets, and on the other side we have a little, almost like a trash chute for the outlet. And so we would just run it through that and get our images. The camera on the bottom connects to your laptop and you're able to take pictures. So then we ran the pictures through our MATLAB code. And so our MATLAB code was able to do a lot. For these pictures, a lot of them were upside down or rotated. So we have to account for that and rotate them back so that when we get the data, it's corresponding with it as well. And we also turned it to grayscale so we can get overall intensity. Grayscale is from 0 to 256. It's just a scale. And if you can look at the intensities over here, you can see that it's relatively low. So we didn't get as much as emission as we would like. So in the future, we want a better light source or a more sensitive camera.
Microfluidics is definitely something that we want to move towards in the future. By using microfluidics, it decreases pipetting, which allows for less chances of errors of occurring. And by using microfluidics, it also allows for a live imaging feed of the mixing of the 2 proteins. And this image right here was the actual mold that we used for our microfluidics strip. As you can see from the ones we were passing around. And with the tubing attached into it, it's shown here in this figure. And we inputted the 2 proteins with a syringe pump, with setting of 10 microliters per minute. And by using syringe pump, it's a more controlled way of inputting the proteins, and it allows for less human pipetting error. And these 2 figures over here were just the microfluidic. We were testing the microfluidic chip in our imaging device. Although we made around like 50 or so of these chips, all of them are still kind of weak. So this is definitely something we want to work on in the future.
Lastly, we will discuss our future work. As previously mentioned by Chris in the last slide, incorporating a fully developed microfluidic device will definitely increase the quality of our results. Also, including automating the whole system will also increase the quality of our results, as well as improving the camera and replacing a camera that we currently have, where a camera that has a better sensor for sensitivity and of course a higher resolution. And improving the position of the light sources to excite our fluorescent protein will definitely also increase the quality of our results. Besides the scientific rewards that our device has, we also have a lot of financial rewards. Currently in the market for research devices, most of the devices that perform similar functions to our device are priced at around $100,000, such as the flex station found in Dr. Liao's lab. However, our device will be priced at a much lower price than that, which will increase the revenue of our product. And we would like to acknowledge the following people. And questions?
Yeah.
Including maybe exposure of testing the samples and taking that also
Yep. In fact, like every time we run the experiment, we're supposed to do it like in complete darkness. Like everything's supposed to be dark. So we actually have a dark room located in MSC, and every run that we had, every experiment that took place in our device was completed in complete darkness. So, are you talking about physically?
The light is aiming down towards the camera, so I don't think it really would affect it very much, because if it's keeping it from scattering out, it's still going to be hitting the wall. It's still going to be hitting your camera. Does that make sense? Yeah.
So the Biocore does not require labeling. That's one of the reasons why I choose. Your system requires labeling, so it's not quite the same in terms of its application.
You're completely right. Right now we do need labeling because we don't have it automated yet. In the future, we'd like to have it so that we have the computer do all the thinking for us and move that wall plate around.
Fluorescent labeling.
Oh, I'm sorry. Sorry, I thought you meant labeling the wall plate.
The materials you're using, you have to chemically label them with fluorescent compound.
Yeah.
But Biocore does not require a fluorescent compound. So that's why it's used.
Any other questions? Questions about the cost of your device? $20.
That was
I think it's about a ticket time, but
we actually didn't come up with this estimate. It was given to us by someone else.
the sales price.
Oh yeah,
The sales.
oh yeah. That's not like that's not a money that it's just the market price.
Okay.
Yeah, it's not a money that the budget that we have.
What's the camera?
It's
It's a CMOS
yeah, we're
camera
using
right
CMOS
now.
camera.
But in the future, we would like to use a CCD or
How much is that seating cost?
That's priced around $400.
but we got ours online from